Another day, another story. Researchers have achieved a groundbreaking milestone by successfully growing mouse embryos in the unique microgravity environment of the International Space Station. This pioneering achievement represents a significant leap forward in our understanding of reproduction in space and has profound implications for the future of human space exploration. In a joint statement by the University of Yamanashi and the National Research Institute RIKEN, this feat was hailed as the first ever study that shows mammals may be able to thrive in space. The experiment, referred to as the world's first experiment that cultured early-stage mammalian embryos under complete microgravity of ISS, signifies a major step towards the possibility of humans reproducing in space. Such an accomplishment would mark a crucial milestone in our journey to becoming a spacefaring species. These momentous results coincide with the preparations for humanity's return to the moon through the Artemis program, which aims to land the first woman and first person of color on the lunar surface by 2025. While the initial lunar occupation may be relatively short, it sets the stage for the establishment of lunar camps, laying the foundation for extended stays in space. This development brings us one step closer to the realization of off-world reproduction, a concept often seen in science fiction. The experiment to grow the first mammalian embryos in space was led by Teruhiko Wakayama, a molecular biologist at the University of Yamanashi's Advanced Biotechnology Center. Their primary objective was to assess whether mammalian fetuses could develop normally in microgravity, and they employed frozen mouse embryos for this mission. These embryos were transported to the ISS aboard a SpaceX Falcon 9 rocket in August 2021. Once aboard the space station, the early-stage mouse embryos were thawed and cultured under microgravity conditions for 4 days by astronauts. Subsequently, these samples were returned to Earth for comparison with mouse embryos cultivated in regular gravity conditions. Remarkably, the study revealed that embryos cultured in microgravity developed into blastocysts, which are clusters of dividing cells formed by fertilized eggs with normal cell numbers. This finding clearly demonstrated that gravity had no significant impact on blastocyst formation and the initial differentiation of mammalian embryos. Furthermore, the research indicated that if allowed, these blastocysts could potentially develop into mouse fetuses and placentas with no significant DNA alterations or changes in gene expression. It's worth noting that the survival rate of the embryos grown on the ISS was somewhat lower than those cultivated on Earth. To progress further, the next crucial step will be to transplant the blastocysts cultured in microgravity into mice to determine if they can give birth, confirming their normalcy. One area of continued investigation is the potential impact of radiation on mammalian embryo growth in space. While the researchers examined this aspect to some extent, they did not specifically address radiation exposure during live embryo cryopreservation and culturing phases. Understanding the effects of radiation is essential as we plan for long-term human stays on the Moon and Mars and contemplate the possibility of colonization. The ability to manipulate and regulate human reproduction has expanded significantly in recent decades, with assisted reproductive technologies such as in vitro fertilization allowing millions of people to start families on their own terms. This technology also holds potential for space colonization, as traditional sexual reproduction in space presents unique challenges due to microgravity's impact on intimacy and sexual function. Initial experiments with animal reproduction in space, including rats, mice, and other species, yielded mixed results, with difficulties in successful pregnancies. However, more recent approaches involve sending freeze-dried sperm, oocytes, or embryos into space, which take up less space and resources on spacecraft. Once at their destination, they can be revived and implanted. Nevertheless, the high levels of cosmic radiation in space pose potential risks to DNA, causing mutations and impairing embryo development. In 2017, researchers sent freeze-dried mouse sperm to the ISS for nearly 10 months. And although the sperm exhibited higher DNA fragmentation, they were able to generate embryos through IVF, and these embryos developed into healthy adult mice, demonstrating the potential of this approach. While some may consider experiments involving human reproduction in space as ethically challenging, they are essential to understand the impact of space travel on human reproduction fully. Recent studies, such as one using reduced gravity conditions to assess human sperm, show that aspects of human reproduction are indeed feasible in space. However, there is still much to learn and accomplish before we can undertake the entire reproductive process beyond our home planet. Thanks for watching.